10 MDCAT Biology MCQS, Staining Techniques, KPK textbook. Here are the 10 multiple-choice questions with full options A, B, C, D, and the correct answers marked clearly. Question 1. What is the primary stain used in the standard gram staining technique? A. Safranin. B. Grams iodine. C. Crystal violet. Correct. D. Carbolfuchsin. Question 2. Which of the following functions as a mordant during the gram staining procedure? A. Grams iodine. Correct. B. 95% ethanol. C. Methylene blue. D. Crystal violet. Question 3. During gram staining, gram-negative bacteria lose the crystal violet iodine complex when treated with which reagent? A. Alcohol or acetone. Correct. B. Distilled water. C. Safranin solution. D. Ammonium oxalate. Question 4. Which staining technique is specifically indicated for identifying mycobacterium species due to the presence of waxy lipids mycolic acid in their cell walls? A. Acid-fast staining. Correct. B. Gram staining. C. Endospore staining. D. Negative staining. Question 5. In negative staining, why does the background become colored while the bacterial cells remain transparent? A. The acidic dye carries a negative charge and is repelled by the negative charge of the bacterial cell surface. Correct. B. The basic dye is actively pumped out of the living bacterial cell via efflux pumps. C. The heat fixation step completely vaporizes the dye inside the cytoplasm. D. The bacterial cell wall lacks lipids necessary to bind the dye particles. Question 6. Pair is correctly matched with the endospore staining technique. A. Malachite green and safranin. Correct. B. Crystal violet and safranin. C. Carbolfuchsin and methylene blue. D. India ink and crystal violet. Question 7. What color do gram-negative bacteria appear under the microscope after a properly completed gram stain? A. Pink or red. Correct. B. Dark purple or blue. C. Bright green. D. Completely colorless. Question 8. What is the primary purpose of heat fixing a bacterial smear on a glass slide before staining? A. To adhere the specimen to the slide and kill the microorganisms. Correct. B. To break open the plasma membrane so internal organelles can absorb dyes. C. To selectively remove lipids from the outer membrane of the cells. D. To ensure the slide becomes hydrophilic enough to repel acidic stains. Question 9. Methylene blue is an example of a basic dye. What property allows basic dyes to bind effectively to bacterial cells? A. They possess a positive charge that is electrostatically attracted to negatively charged cell components. Correct. B. They are highly hydrophobic and dissolve readily inside lipid bilayers. C. They form covalent bonds, specifically with the peptidoglycan molecules. D. They react chemically with the silicon molecules on the glass surface. Question 10. Which specialized staining method requires a mordant like tannic acid to artificially build up the thickness of hair-like structures until they become visible under a light microscope? A. Flagella staining. Correct. B. Capsule staining. C. Simple staining. D. Acid-fast staining.